Bonjour, mes amis. It is wonderful to have you and your guts here with us this evening. Today, we are going to be looking at why gut health and gut health advice is never going to be a one size fits all. With over 100 trillion little critters living in your gut, the way you process food depends not only on that biome, but also the genes that inform the gut and the biome. So, in this webinar, we're going to be uncovering different genetic variants that will be influencing that. So, take this as your cue to start pulling out the paperwork and the apps that you have that help inform your genetic understanding. So that might mean pulling up the DNA Way app. We're going to put a link to that in the chat, or maybe that means jumping into your portal. So that you can be ready, I'd encourage you to go to our diet and nutrition section, and there should be a summary page that lists out all the genes. Today, and I'll write this in the chat so you have access to it, we're going to be looking at MCM6, FUT2, and Amy1, amongst a few others. And so all of these genes that we explore are going to be things that influence how we tolerate or maybe even have an affinity towards certain foods. It's going to explain why healthy foods might work really well for one person, and then for the next, they become very problematic. So we're going to look at, again, how the DNA 360 can inform what is truly healthy for you, as opposed to that crass label of healthy food versus not healthy food. And we're going to also look at what some advanced biome testing can say not only about your gut biome, but the state of your full body's health. I'm excited to do this with you. I'm going to be doing it in conjunction this evening with our most wonderful Dr. Laura Varden. Medal, welcome to the stage. How are you doing?
Well, hello and good evening. I'm doing well. Thank you for asking.
Yay!
It's so nice to be able to host a webinar with you 2 nights in a row. This is
I
wonderful.
know,
I
it
feel
is
so honored.
delightful. It is delightful. Yeah. And so, audience, Dr. Varden and I show up to the end of supporting your understanding, supporting what you're seeking to know. And so, one of the ways we look into that is through the poll questions. So, I actually want to kick off this webinar with those very poll questions so that we can refine what we talk about specifically to what you're hoping to learn. So, Vanny, Katie, perfect. Thank you guys. So, audience, the first question that we asked you is, how familiar are you with your gut biome? And about 70% of the audience, a sizable majority, saying they understand the basics. And then we have a happy 20% saying that they're already tracking and testing their biome. I'm really curious, for those that are tracking and testing, what are the insights that you've gotten from that, and have they actually helped you? That question is always very interesting to me because I think a lot of people do the testing and it doesn't always translate into help. And I want to make sure we unravel the why behind that. So, audience, if you're someone that's tested and you've played with it, toss it into the chat. I want to know your experience there. The next question we asked is, which area of gut health do you struggle with the most? And there's a happy little smattering. I would say the top three are going to be bloating, food sensitivity, and irregular digestion. So that can that's the fluctuation picture between going from constipation to diarrhea and maybe a little bit in between, but kind of that very uncomfortable picture as opposed to those formed bowel movements that we all want. The next question we asked is, which diet advice confuses you the most? And the most frequent offender here is going to be probiotics. Are they worth it, or are they just hype? I love this question. I'm so passionate and sometimes even dispassionate around probiotics. And I'm grateful in particular when we talk about biome testing, how that can further refine the specifics to you. Because the answer, I mean, as you would come to expect from us, is never a clear yes or no. It's always going to be, it depends on you. The next thing that people are a little bit confused about is dairy, good or bad? And within genes, we're going to start tackling that question. The next question we had is asking, if you could personalize your nutrition to improve one outcome, what would it be? And the biggest winner here is going to be about 40% of the audience is looking for some long-term disease prevention, just as my cat is looking for long-term exposure to you all. She says, good evening. And the next thing that people are looking for is a little bit more energy and better digestion. So, again, we'll touch on all of those as we go through genes and really that preventative, personalized lens. The last question we asked is, what are you hoping to gain most from this session? And the biggest answer here is going to be a better understanding of the role of genetics in digestion. And so I'm glad we've picked the genes that we have because those are really going to give some insight into your digestion there. Yeah. Okay. And so let us start jumping into things, Dr. Varden. Are you ready? Do you have your engine all
I
revved?
am. Absolutely. And we've got some good comments coming in
Mm,
about different tests that people have
mm,
taken, what they've gotten out of it. We have Rachel saying that only in the beginning phase of testing, she's got a lot of food sensitivities showing so far. Could be leaky gut, but she has more testing to go. And Nina's
mm,
saying bark half is the best for microbiome and constipation. So I guess that was just a comment. Joyce saying the most significant finding on her gut test was that she has low commensal bacteria. So
mm.
she's working on that. Lisa
Amazing.
saying that low pancreatic enzymes. So she takes enzymes prior to eating, adds vinegar to water prior to her meals, but doesn't know why the enzymes are so low. It's a great
Mm,
question, going to the root cause.
yeah.
Um, and different people, Lisa saying she has confirmed leaky gut. So, but the real thing is, okay, so you have this information, and it sounds like a couple of you are saying, oh, well, you know what you're going to be doing, some of these next steps to take to help, either heal leaky gut or figure out what the true problem is so that way you can focus on where to place therapies or place your focus. And that's really what it comes down to. It's like knowing the information can be great, but what do you do about it? What are those action steps to really start making changes that can
Hmm.
start healing the gut, start optimizing your body? Because your gut plays so many factors, so many important, impactful aspects in your body, in your brain, that gut brain connection, the fact that it really is the mainstay of your immune system. How well are you absorbing the nutrients that you're taking in? So all of these things very important for gut health. And of course, genes can play a factor in that.
So why don't we dive right into those genes? Because I think the audience is really looking for two things, right? Like how do we personalize the health? But first, let's start with the genes because that serves as the foundation for helping to understand what might we be sensitive to, what's going to shift a healthy food to a non-healthy food, right?
Right.
Yeah. So let us start, audience, again, through your 360, have the diet and nutrition section open. We're going to be diving into MCM6. Okay. Um, and so this is a gene that is going to look at, do you create sufficient amounts, strong amounts of an enzyme that helps break down lactose? So when we ask the question of dairy, right? One of the principal things that can bother a lot of people that causes a lot of bloating in people is going to be inability to digest this sugar. Okay. Um, let's talk about the how of that because it really involves the gut biome. So we eat that beautiful yogurt, that ice cream, whatever it is, that cappuccino with the milk in it. Um, and there is going to be some lactose in it. That is a sugar very specific to dairy, to milk, goes into your gut. And if you don't have that enzyme to break that up, the bacteria in your gut are like, cool, she's not eating it, so I'm going to eat it. And what happens is when they munch that up, it creates a lot of gas. And that gas is going to lend to a lot of bloating. That gas is also going to end to kind of, that like poppy sensation. That's all the gas that's being created, those bubbles, and then they're going to pop. And ultimately, that's going to bother the GI. That is going to create a sense of urgency needing to go to the bathroom. That's going to create a little bit of pain from that gas and bloating. And that's also going to contribute a lot to loose stool. Because if you have this disruption going on in the gut, the body's like, cool, we need to get rid of this. So, knowing your presentation of MCM6 really helps you understand, do I have capacity, especially in later years, right? As babies, we tend to be decent with this enzyme independent of genes. We're more talking as you get up there in age, right? Anything past 20, really. Um, do you have the ability to instinctively digest this? So, looking at your test can give a little bit of insight there. Dr. Varden, do you want to talk through one of my more favored diet nutrition genes? Do you want to talk through Amy1?
Absolutely. So Amy 1 is really interesting because it first of all, it deals with starch digestion and its efficiency and works into blood sugar response. So with Amy 1, which is short for amylase 1, which is the enzyme in the saliva in the mouth that helps to break down the carbohydrates. And with this gene, depending on the allele or the variant that you have, it will dictate how many copies of the gene you have. So, for example, if you have a T allele, that says that you have more copies, an A allele, less copies. And you can have anywhere from 2 to 17 copies. Obviously, the more copies you have, the faster you break down starch. So, research has shown that those with more Amy 1 copies also develop different gut microbiome profiles, especially more in the Rhamnococcus family, that the bacteria that assist in breaking down resistant starches. And this really co-evolved mechanism makes starch digestion really a team effort between genes and the microbes. And you can think about the Amy 1, if starch digestion, say, were a kitchen job, okay? So Amy 1 can represent the number of helpers you have. The more helpers, the faster and smoother your carbohydrate meal gets prepped. So really that's where it comes in. And this is something that can influence how well you may take in different types of starches, if how well you might be able to digest some of those breads and pastas and other things, or whether it gives you some GI upset.
Yeah. And it also shifts, I think, well, I know it also shifts your experience of food after the fact. So if you're someone with a poor Amy 1 presentation and you have all the sugars, right? You sit down, you have that fabulous pasta, pure, very simple carbohydrate. In the moment, you're probably very happy. But what's happening inside of the body, because you're not breaking that down optimally, you actually get a bigger blood sugar spike. And because of that spike, it also means you're going to drop just as quickly. And so when people drop, there's an element of individual biology. Some people are going to experience that as an energy crash, right? They get the itis. They've just eaten and now they're like, "Ooh, okay, bedtime." And then other people are going to experience that a little bit as anxiety because sometimes that's how the brain interprets it. So people with a suboptimal presentation here, they tend to also have a different relationship with carbohydrate in that they want it more because they're delicious, but then they don't necessarily respond as optimally as they might want to. So something to keep in mind, not only in the digestion side of things, but the metabolism side of things too.
Yeah. And I mean, when we're talking about the number, the copies that you have, again, that can range between that 2 to 17. So depending on what type of combination that you have, because Janet had a question, what if my Amy 1 is an AT? Well, then you're somewhere in the middle,
Mhm.
of that. Exactly how many? Not sure. We'd actually have to go in and take a look. But yeah, you're somewhere in the middle, but definitely on that lower side. So think again, go back to the kitchen helpers. You, instead of having 17 helpers, you may only have, 9, somewhere around there.
Yeah. We look at a SNP that really influences
Mhm.
the secretion amount. So it is a SNP that we look at, not a copy number variation, but it's very specific
Mhm.
to how much is coming out. Yeah. Let's talk about FUT2. This is a happy little merging of how body supports biome. So, FUT2, a lot of people have heard this in terms of secretor status. Do you secrete this specific, or do you present with this specific enzyme? And I'll offer a little bit of an analogy. The people who are secretors, the people who exhibit this enzyme, they're really, really good at throwing a party for gut bacteria. So, what do you need to throw a party for gut bacteria? First of all, you want a really warm and welcoming environment. You want somewhere for the bacteria to cling on to. And to an extent, you want something that's going to be feeding. You, Food's very important. No one goes to a party unless there's good food. And so if you're a secretor, you are someone that has afforded and created a really good party, and your gut bacteria wants to be there. There are points with which that bacteria can grab on and set up camp. There's places that that bacteria can go to and be fed really well. So, knowing your FUT2 status really will speak to how instinctively, how strongly, how happily can you plan a party for your bacteria. And when we look into the research, when we look into comparing people, even comparing babies, audience, this is kind of cool. If we look at babies that are a few weeks outside of their mama after birth, we're interestingly able to tell who is an FUT2 secretor compared to who is not, simply by a biome test. Okay, that's how influential it is in setting up that bacterial party. Now, there's good and bad to this, right? The good is if you're setting up a good party, right, you have better diversity, better outcomes in that ability to regulate your biome. The tricky of that sometimes is you sometimes get bad bacteria jumping onto those placeholders, those connection points that I mentioned. And so you're a little more prone to foodborne illness, right? So those that don't have it, well, I wouldn't say that's ideal per se, they're at least a little more safeguarded from foodborne illnesses. I do not suggest a science test. Please do not try that out. But research has shown, especially like when we talk about cruises and things like that, those that are not secretors tend to be a little more immune to that sort of exposure. So that is what FUT2 tells us. Dr. Verdon, I know you like the next gene. Do you want to jump into that one?
Are you talking about the SLC23A1?
Yes, ma'am. Look at that little head bob.
All right. So this is one that we don't normally talk about, but it is very important. And this is actually a solute carrier for vitamin C. So it transports vitamin C and works in with absorption because it's the main transporter that draws vitamin C into your cells. Vitamin C, of course, is a very important antioxidant. It has numerous functions, but the gene itself, depending on if you have a variant, for example, if you have a G allele, that is an optimal presentation would be like a GG. If you have an A in there, an A is an apple, okay? This is where you actually start having slower transport, you can have reduced blood and tissue vitamin C levels, actually up to 50%. So, you know, for people who are dysregulated in this gene, you may need higher dietary or supplemental vitamin C for optimal
Mhm.
antioxidant production and immune resilience. Because again, vitamin C very, very important for
Yeah.
for the immune system. So I love analogies, and I love the one Dr. Kosterman gave for the FUT2. So thinking about an analogy for vitamin C, if you imagine that gene as your vitamin C delivery truck with a variant related slowdown, your drivers might need reinforcement from more vitamin C rich foods or supplements to keep the whole system running smoothly. So, this is something to check in your reports. It would be in your diet and nutrition report. And you can take a look to see if you might need a little extra supplementation of vitamin C rich foods or straight up supplements.
Yeah, I'm seeing some questions asking where some of these genes are, so I'll be a bit more specific. If you're looking for FUT2, within diet and nutrition, there will be a sub-report that is called the microbiome. You'll see your FUT2 there. If you are looking for SLC23A1, you will find that either in your system summary, but also in the section called micronutrients. Okay, and that's all within diet and nutrition. These are just little reports within diet and nutrition. Yeah. So I think by exploring, or at least the few genes that Dr. Varden have listed, we really are capturing how there can be some really individualized responses to different foods, to different diets, and why this idea of kind of the prescription of one diet for all isn't always going to be healthy or helpful. Sorry, helpful. There we go. So I want to explore, or at least Dr. Varden, I love it if we could explore a little bit more how the microbiome is really shaped by what we eat and influential to how we eat and how we absorb.
Absolutely.
Yeah.
I mean, you really need to understand that when it comes to your genes, yes, they play an important part. Like Dr. Kosterman said, in babies, you can immediately tell just through stool samples, those that have FUT2 dysregulation, whether they're secretors or non-secretors. But again, genes only are part of the story. Your microbiome is the other half. And your microbiome is diverse. Well, I mean, at least we want it to be very diverse,
Yeah.
dynamic, and is directly shaped by what you eat.
Hmm.
So, you should think of it like you have an army and you have different factions and some that fight each other, and there's some good guys and bad guys, and yeah, some bad guys live in there, and it just is what it is. But generally, you want to keep the bad guys in smaller numbers, so the good guys are kind of patrolling and making sure that they're under control. Well, all of your troops and all of they need to eat. Well, what if you're only giving food that the bad guys like? Guess who's going to get stronger and larger in number? Yeah, the bad guys. Okay. So if you're eating a lot of sugary foods, if you are eating a lot of processed foods, this isn't going to build up the good gut bacteria to make them strong, to feed them. So you want to make sure that you are choosing foods that feed the good gut bacteria, that are feeding those keystone species like your lactobacillus, your bifidobacterium,
Yeah.
Akkermansia, all of these wonderful phylums, these family of bacterium that are there to support good gut health, good immune system, that are producing these what we call postbiotics, which
Hmm.
are generally short chain fatty acids. Butyrate, is a big one, that helps to give information for the brain, for the body in a good way. Instead of feeding, like lots of sugar, what can increase? Candida and
Yeah.
other, bacterium that can cause issues.
Yeah.
So it's very, very important that we are feeding the good guys.
Yeah.
And this also lends to that foods that heal versus foods that are harming.
Yeah.
And that microbiome and genes obviously work together to play a part in that.
Yeah. And different populations of bacteria as well will influence how you receive. There's certain populations of bacteria, for example, that have a really big affinity for fat. And so when you consume large fat meals and this population starts to grow, you actually start to extract more energy from your food, meaning that not all calories are alike, that your biome will influence how many calories your food has. So, what do I mean by this? If you have a population that is set up with a lot of Firmicutes, which is a family that is fat affiliated, if you have a higher proportion of that, if you take 100 calories in, you actually have more access to that 100 calories compared to someone with a lower population of Firmicutes who takes in that same 100 calories and maybe only 80 actually translates and is actually brought into the body. So to me, it's really interesting that the different populations not only influence immune, not only influence brain health, inflammation, all that fun stuff, it also influences what we're taking from our food, how our body has access to and can receive that different food. Yeah. And I think um, this is where intelligent gut testing becomes really, really handy. And this is why I say this, audience, when a lot of you had mentioned I've taken these sorts of tests, and my curiosity was always, well, what did you do with those answers? Right. And sometimes, especially if there's something very obviously wrong. Maybe there's an overgrowth of something opportunistic. Maybe there's something more pathogenic. The answer to what to do with that is obvious. Sometimes, though, especially if you kind of get a middle of the road test, or sometimes if you're seeing one population high, it isn't always helpful. And I found this personally as a practicing naturopath, in that I would see a lot of these tests and I would say, these tests are focusing on a few species, but it's really difficult to get a full picture of what's going on in the gut. Actually, I'll be honest, for a large portion, the beginning of my career, I shied away from a lot of gut testing, not because I didn't think it wasn't powerful, but because I couldn't see the translation. How can we go from 100 trillion, or whatever that obscenely large number is, of little critters in the gut, and then boil it down to maybe 100 things that I'm seeing on a piece of paper? It didn't make sense to me. But this is where the beautiful of AI and the beautiful of big data comes in,
Mm.
right? So when we look at testing like our Gut 360 Premium, what it does is not only does it, and I love that I get to say this so dismissively, not only does it index everything that all the bacteria in your gut, which honestly is a feat by itself, but not only does it index all the bacteria in the gut, what's more important to me is that it analyzes the different patterns, because audience, that is what we're learning is actually the sweet spot and the goal when it comes to biome analysis, right? Short of a very overt pathogen, our gut is a relationship, it's a community, it's made up of different characters, and we want to know how those characters are in population and how they are working together. So when you have large AI models, you're able to look at that patterning. Computers can look at it. I can't. Dr. Varden can't. We're humans. We don't have that capacity. But the computer can tell us, hey, you have a pattern that we see often in depression. Hey, you have a pattern we often see in IBS or IBD. Hey, you have a pattern that we see in dementia or Alzheimer's. And so that is able to say, this is what's going on in the gut. This is how the gut is informing your body. We know the gut informs mental health. We know the gut informs your skin. We know the gut informs inflammation and immune, right? It helps us understand the patterns that are informing the body, and more importantly, what to do about it, right? This is where that probiotic question comes in, because I would say, are probiotics good? They can be when they're treated like medicine and they're addressing a specific issue within the gut. And that's what our testing can help elucidate, right? It's saying, based on this pattern, based on a lack or a surplus of a certain population, you can adjust it using this. And with that precision, you actually don't disrupt the biome, you're helping it. How many times an audience, I'm sure someone on here has felt this before, where they've taken a probiotic and it felt like World War III in their gut. you know, it's that,
Put a one in the chat if that's happened
yeah.
to you. You take a probiotic and you're like, what the heck did I just do? And
yeah.
I
And
would love to see. There we go.
yeah.
Yeah.
Yeah, yeah,
Yeah.
we're seeing a bunch of ones right now, right? And
Yeah.
that's not because probiotics are bad. That's because the probiotic you took was mismatched to the pattern in your gut, And then they went like this. I can't, Lord knows I can't fight, but they tried to beat one another up like this. That's how I fight. Right. And that's why
Really?
it felt horrible inside the gut. And so when someone asks me the question, are probiotics are good? I'm like, that's not really a question. The question is, what is appropriate for your gut right now? Yeah.
Exactly. And circling back to your genetics, to the foods that you
Hmm.
eat, to, looking at your current microbiome, what the landscape looks like. There was actually a study where the researchers followed, I think, like 800 people, and they
Mm.
tracked their food intake, their blood sugar, their genetics, and microbiome. And you know what they found? They found that the same food caused totally different blood sugar
Mhm.
spikes depending on each person's genes and microbiome.
Mhm. Yeah.
So that right there, you really need to take away from that. When distilling it down, there's no universal healthy food. Okay. Whether a food helps or harms really depends on your unique biology, your current microbial diversity and composition. Because even, you sitting there, what it is now, you make some changes. Say you're having some issues, you're getting blood sugar spikes and things are a bit out of whack. You make some changes, change up your diet, maybe take very specific precision, because hopefully you know which probiotics you need. You take those, give it a handful of months, take your test again. Oh, hey, all of a sudden your blood sugar is not spiking as much. Your gut composition has changed. The foods that you're eating are now affecting you better or differently than they were. What hasn't changed? Your genes, but you're changing lifestyle, you're changing diet. You have
Mhm.
efficiently changed the composition and the population of your gut biome. Yes, it can happen. Does it happen like that? No, not necessarily. It really takes someone who understands and knows how to kind of start nudging these changes to start
Mhm.
having very precise and specific changes in your
Mhm.
lifestyle, in your food choices. And that's what we do. We look at
Mhm.
all of these things. So if you're having issues, please, you know, let us know. Give us, a call. Speak with, one of our team, and get a consultation. Work with one of us, and we can help you along that path for a
Mhm.
more precise therapy and path. Putting all these pieces together.
Yeah. All right. Dr.
Oh,
Varden, do you
oh,
want?
and
Oh,
wait a
oh,
minute.
oh,
One thing
that's an
you
excited face.
forgot. Yes. Um, for the Gut 360 Premium that Dr. Kosterman is
Oh,
talking about,
yeah. I'm glad you're
we
smart.
have a 10% off discount code. If you limited time, Vanny will put it in the chat. But if you use gut 10 at checkout, you'll get 10% off that test. So highly recommend. Again, test, don't guess. Know what you have got going on, so you know
Mm,
how more precisely to start nudging and pushing and making changes the way you want.
yeah. 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 It's an incredible meeting of technology and action. Yeah, I'm excited for it. Yeah, and I'm excited what it can offer people because I think gut testing is important, but the interpretation all the more. Yeah. Dr. Varden, do you want us to use some myth busting?
Oh, absolutely.
All right, let's start with the first myth. I have down here dairy is always bad. Oui ou non?
I love my ice cream. Dairy's not bad. Um,
Yeah.
this is this is circling back around to your gene, your MCM6 gene. dealing with lactose that we talked about earlier. So, are you prone for lactose intolerance, partial lactose intolerance? It really, it's really interesting because I've had several clients uh, say they're partially lactose intolerant or they are genetically predisposed or as lactose intolerant. Okay, they have a GG presentation. Okay, and yet they're like, oh well, I eat dairy and I don't have any major issues. And it's like, oh, okay, very interesting. Well, you wonder why. Well, it can be that throughout maybe they have trained their bodies and their gut biome to be able to handle that. But the interesting thing is a lot of times I'll ask them, um, do you have any type of sensitivities or any type of reactions? And they're like, I don't know. And I said, well, do you get phlegmy? Or after you eat it, and they're like, oh yeah, actually I do. Well, that's part of a lactose or dairy sensitivity. And honestly, I get that a little bit too. But is it bad enough for me to stop eating ice cream and dairy? No. So it's going in knowing, informed, saying, okay, these are my genes. How bad are symptoms that I may or may not have? And is it worth removing totally? Now, do I have dairy every day? Is it a major part of my diet? No, because I
Mhm.
also kind of know how I feel afterward. It may taste great going down, but it may not be the best after.
Yeah.
So again, it's what's going on with you. But
Yeah.
dairy is not always bad, and also products thereof, yogurts,
Yeah.
great for probiotics. So there's so many different things that you need to take into account, but trying to
Hmm.
distill it down just to a phrase saying dairy is bad, no.
Yeah. And I think Angelil, if I'm saying that correctly, is also pulling out a nuance that it isn't always about just the sugar.
Right?
Within dairy, there's considerations of protein.
Proteins.
Some people might be sensitive to casein or whey, and then there's some people that they produce some immune byproducts that aren't favorable. So there's an element of further personalization, but when we focus on the MCM6, right, that really speaks to that ability to digest the sugar, which is one of the first things that can disrupt our experience of wellness and the gut biome. Yeah.
There's always rabbit holes to go down for each one of these, but we need to keep ourselves reined in. So
Yeah. Yeah. All right. The next one is this one's going to make me very sad. All right. The next myth is gluten-free is always healthier. And the answer to this, like, so audience, I'm celiac. It gets me very angry that I cannot eat my deity, which is a croissant, but here we are. Audience, the answer to this is actually no. And before I start with genes, I want to start with a study. There was a study done 10 years ago, kind of at the beginning of the gluten-free craze, and it looked at people who did not have genetic or health reasons to take out gluten. Okay, so take those two things off the table. There's no genetic reason or health reason why they should be taking out gluten. And so these people took out gluten because it was trendy and they were like, maybe it's healthier. And they looked at these people's biomes compared to people who also had similar health profiles but did not take out gluten. Okay. And the people who had taken out gluten had a worse biome picture after it was either 30 or 60 days. I forget exactly how long they followed people, but after 30 or 60 days, people who had taken out gluten had a worse biome picture. Now, why is that? And this is really important for anyone who is considering to take out the deity that is gluten. It is because oftentimes when people take it out, they put in replacement products that are high in sugar and high in simple carbohydrate and lack oftentimes fiber, right? They're not taking out that piece of bread and replacing it with some sweet potato. What they're doing is they're taking out that piece of bread and replacing it with some gluten-free bread that does not have the fiber and some of the protein and amongst other things that can be really good for the gut. Okay. So while I think there's a lot of genetic considerations, if you jump into your Gut 360, if you look in the diet nutrition section, if you look at the carbohydrate there, we'll talk to you. We will let you know, do your genes predisposed towards celiac or predisposed towards gluten sensitivity? And maybe encoded in you, unfortunately, is this statement of gluten isn't good for you. But for those that don't have that, for those that don't have a health picture, gluten-free is not always healthier, right? So vive le croissant is how I'm going to end that one. All right. Dr. Varden, do you want to jump
Yes.
into the idea of more fiber is always better?
Absolutely. And here's the thing that unfortunately, for some individuals, especially those with FUT2 variants that Dr. Kosterman so beautifully explained, the secretors versus the non-secretors, or inflammation prone genetic profiles. Excessive fermentable fibers can actually worsen gut symptoms instead of improving them. And really, what ends up happening, for example, with the FUT2 gene, remember how she was talking about how it encodes that enzyme, okay? Because it actually is a fucosal transferase enzyme that determines your secretor status. So whether you secrete a blood group antigen, okay, which this is what we call a fructose, it's a sugar based, okay? It's a fructose based sugar into mucus that lines the gut. All right. So these sugars serve as food for beneficial microbes like bifidobacteria, lactose bacillus. All right. And really, when it comes down to, if you remember me talking about how the bacterium, a postbiotic, okay, short chain fatty acids such as butyrate, they depend on healthier fiber fermentation by beneficial microbes. Non-secretors often have less efficient fermentation and weaker short chain fatty acid production. So in those individuals, high fiber diets, especially those high in fermentable oligosaccharides or resistant starch, it can overwhelm the imbalanced microbiome that can trigger gas, bloating, inflammation. So, it's too bad that most dietary advice really glorifies fiber as universally beneficial for bowel health and microbiome diversity. But for those non-secretors or individuals, again, with inflammation prone genetic variants, certain fibers can definitely exacerbate problems rather than help them.
Mm.
So,
Yeah. Yeah. All right. And our last myth, I think, before we start perhaps transitioning into a few questions and answers, because our audience has quite a few of those this evening. The last one is an idea of probiotics work for everyone. Be the Oprah of probiotics, one for you and you and you. And I think we explored already, especially through a lot of audience experience, that that's not the case. I think there's definitely a time and a place and a purpose for probiotic, but I think the idea that you can go to a store, pick anything off the shelf, and it's going to be great for you is thankfully coming to a close. We're looking a lot more at precision medicine, which means understanding what's going on in your gut, knowing what that pattern is, right? And then supporting the pattern intelligently through specific actors, as opposed to a very generalized thing, right? It's like stepping away from a multivitamin and going to specific vitamins that your body needs. You'd be stepping away from a general kind of off the shelf idea to more specific strains that support you. And that's where the beautiful of big data and artificial intelligence comes in, right? That larger mapping that meager minds can't always get, or human minds, I guess, can't get our heads wrapped around. Yeah, yeah. Dr. Varden, before we transition into our questions, um,
mhm.
is there anything you want to offer to the audience, especially in terms of we've talked a lot about different genes that influence how we receive our food, how we bring food into the body. We've talked a lot about bacteria. Any little tidbits, any clinical pearls, any Dr. Varden bombs that you want to offer the audience of how they can translate, how they can take tonight's webinar and enliven it tomorrow and help it, have it applied to their health.
I I still like the test, don't guess.
Yeah.
If you are having problems, then actually know what's going on.
Hmm.
I mean, that is so important to really spotlight what is going on. I mean, I remember, I felt fine. I remember last year I did our gut 360, and lo and behold, I had high E. coli. And I
Mm.
was like, oh, if I didn't know that, I could have ended up having it get worse and having issues, further downstream. So it really enlightened me going, oh, okay. And I'm also low in my Akkermansia. I need to, make sure that I had more, I'm trying to remember if it was bifidobacterium. So what
Mm.
that allowed me to do was to take steps into shifting some of the foods that I was eating, and also taking specific probiotics that would help to increase the particular keystone species that I was low or missing on.
Mhm.
And then also helping to shift, to kind of crowd out the uh, the E. coli, these pathogenic species.
Mhm.
So, I mean, I did it because, well, I work for the DNA company, and, I wanted to, use one of our new tests. So I found that very insightful.
Mm.
And again, it's important that you just don't throw spaghetti at a wall, just taking any old probiotic,
Mm.
eating these foods and going, oh, my stomach, or, just the bloating or gassy. And, because you could have species that are, big methane producers, or are other type that it's like, how do you know unless you actually do some tests for it? So that way you can have more targeted therapies. So
Mhm.
that's really where it comes down to. If you're having an issue, to figure out what it is, test, don't guess.
Yeah. J'adore. Merci, Madame. Let us question, let us answer. Yeah. Our
Yes,
first
we have a
question,
lot of wonderful
we
questions.
do. We do. The first one I'm seeing is from Sonia, and they're asking, are pickled beets good for the gut? Yeah. My thought there, Sonia, is they can be. They can be. I think I'll be honest, outside of a clinical plan, fermented foods and soluble fibers tend to be my number one approach towards seeding and maintaining and supporting good bacteria. And so pickled beets absolutely fits into that, assuming, you have that live culture in there. And beets can be so delightful for many other things,
Mm.
especially for nitric oxide, right? So I think they can be, but there are going to be some people with a bit of a dysbiotic presentation in their gut where that's not going to be favorable. Or some people may be a little more histamine sensitive. So I think in general, yes, but we always have to peer into the specifics of you. And that can be what working with a provider and getting some testing can help elucidate for you. So just a thought there for you, Sonia. Um, let's see. The
Yeah.
next question that we have, oh my, this might be for me, Dr. Varden. It's from
Yeah,
Sheila.
I know. I saw that. I'm like, I'm going to leave that one for you.
Sheila's asking the question of, I would like to know if you have any hacks of how I can eat a yummy croissant. I love the choice of food there, Sheila. Even if once in a while, if I'm predisposed towards gluten sensitivity and predisposed towards celiac. Um, so within our testing, we look at the HLA, right? And that's a gene that will influence both gluten sensitivity and separately celiac. If your test comes back and it says, hey, you have some SNPs that predispose you towards celiac, when we look at the numbers between who has those SNPs and who actually gets celiac, meaning they've had a test, they've had that biopsy that confirms gluten, causes havoc within the body, it's only about 30 to 40% of people that translate. Now, remember, celiac is an autoimmune condition. There's a lot of variables that go into that. So there's going to be a portion of people, right? This may be you, Sheila, I don't know, that if their health is otherwise really well founded and they don't have those risks for autoimmune conditions, right? Um, that means that maybe gluten can become part of their diet, right? I would consider, I think there's a lot of variables, and I'd work with a provider to understand this, because you also have to look at, when we talk about wheat, some of the accoutrements, right? Those glycophosphates, et cetera, tend to be very problematic for certain genetic profiles. So I think it needs to be evaluated in whole. But for some people, it's an option. And for those that are, and especially those that are a little bit more concerned about it, I would really encourage some digestive enzymes that couple alongside that. That, as well as considering things like sourdough bread and other traditional preparations that break down some of that protein, right? There's many companies out there that offer gluten proteins, gluten digestive enzymes. Two of my favorite, one comes from BioOptimizer. I think it's called like Gluten Guardian. And then there's another one by Enzymatica. Um, that's like there's gluten in the name. I forget exactly what the product is, but both of those are very specific. And so if you're someone that likes to walk on the wild side and you've talked with a provider, that is something that you can be considering, and feel free to tell me how glorious it is, and I'll be crying from the sidelines. All right. Dr. Varden, do you want to grab the next question for us?
Let's
Oh,
see. Ooh.
yeah.
The next question from Shelley.
Hmm.
Uh, after, yeah, let me just do that. Uh, she says, after her GI map test, her functional medicine provider suggested she get Lyme tested, Lyme disease. And it does look like she may have it. Does the DNA
Hmm.
company help with Lyme detox while also taking genetics and perimenopause into consideration? And the answer would be yes. We do come at it from a functional genomics lens, looking at how your detox pathways are, and also at a holistic and integrative approach. Actually, Dr. Kosterman had a wonderful conversation with
Hmm.
Dr. Horowitz, who is a specialist in Lyme disease. He's out of New
Hmm.
York City. And he gave wonderful information, and it really works
Hmm.
along what we do. And so, absolutely. And we deal with women in perimenopause, going through menopause, postmenopausal, and obviously, of course, genetics. So, but
Hmm.
definitely, please reach out, and one of our team members can talk to you and help you
Yeah.
decide on how you want to move forward in a possible program.
Yeah. Um, Vanny or Katie, if you want to toss the email that Shelley can engage with, it's ceo@thednacompany.com. You'll go directly to Tracy to discuss that. All right. Jolene has our next question, and Jolene is asking in regards to food sensitivity. And in essence, she's saying that, she identified she had a food sensitivity. She took that food out. Um, and the question really is, can you restore your gut such that you may not be sensitive to it after a while? If you do food sensitivity testing, does that mean that's a forevermore thing? And then, should you be retesting before reintroducing it, and things along that line, right? So, Jolene, what I would say is not everything is set in stone. Why do I say this, right? We talked about that one bacterial family that has a huge affinity for fat. Right. So if you continually feed that, that's going to increase that population. That's going to increase the calories that your body has access to based off of that. If you shift your diet, if you remove some of that fat, that population will naturally deplete, and that's going to shift how you're extracting calories from food. The same can be said both within the biome and then immune sensitivity to certain foods, right? Sometimes what happens when we have a food sensitivity, and we'll use eggs for example, it's not so much that you are sensitive to eggs in general. It's just that there is so much going on, inflammation and immune wise within the gut. The gut is throwing up false alarms at foods that, if you calm that inflammation and that dysregulation down, if you support the biome better, if you solve for that leaky gut, you would not be sensitive to it. right? Now, the very specific question about how do we do the testing? At what point do we introduce? Can something, a sensitivity go away? Those are things you really do need to explore with a clinician, right? The exploration is how do we resolve that inflammation? Like, how do we identify what isn't working? How do we resolve it? And then what is the plan to see if we can bring it back in? Because sometimes a food sensitivity is straight up that. It's not something that you're going to be able to reintroduce. But a lot of the time, with the proper care and the proper gut healing, it can be things you can bring back in, not necessarily en masse, right? If it's eggs, you may not be able to eat eggs every day, all the day, right? But it can be something you perhaps bring back in. So I'd really encourage working with a clinician because that's a bit more of a nuanced care plan. Yeah. All right, Dr. Biden, I got one for you. Marianne is
Yes.
asking, does Amy 1 affect vegetable digestion? And if so, which veggies?
Well, actually, yes. Amy one, remember, it breaks down starches. Okay, so obviously it's going to influence those foods that are rich in starch. So when we're talking about starchy vegetables, we are looking at things like potatoes and corn, green peas, winter squash. Carrots. Got any others that you can think of? Um, I would say
Yeah.
really it works into the amount of starch. Now, obviously, carrots have moderate starch content,
Mm-hmm.
um, you know, but really when it comes to non-starchy vegetables like the leafy greens, broccoli, cauliflower, zucchini, peppers, tomatoes, they contain
Yeah.
little to no starch, and so therefore are not notably affected by that AME1 gene, or
Mm-hmm.
variations. So just
Yeah.
think of starch, higher starch, AME1 is going to
Yeah.
be affecting those particular vegetables.
Yeah. Good answer there. I apologize. I was trying to prepare for the next question. Yeah, but I also like to think like it's really the refined foods that become most problematic with Amy 1. Yeah. All right. Cheryl has our next question, and Cheryl is asking if vitamins are not being absorbed well into the cell despite supplementation, which gene is the problem? So, Cheryl, what we explored this evening is that it's going to be different genes for different minerals and vitamins. So, for example, Dr. Varden talked through SLC23A1, and that influences vitamin C. We look at another gene, oh no, hold on. Oh my gosh, my brain is farting. SLC30A8. I got that
That
right.
is correct
Yeah.
for zinc.
Yes.
Yep.
Yes, that one looks at zinc, right? We also have a suite of genes that look at vitamin D. And then FUT2, we didn't get too deep into this, but that also influences your ability to absorb B12. So it's a little bit of a tricky question, Cheryl, in that different genes will have different influences. And so I'd encourage that you go into your diet and nutrition report and explore some of the implications there, because that will have a bit of a summary. Okay.
And that actually dovetails nicely into Rachel's question, because she was saying, could you go over which alleles are optimal for the FUT2 and MCM6? Because she said we sort of stayed general on those two,
Mhm.
without really diving into the alleles, because what you just said with the FUT2, that working into B12 absorption into the gut. Well, if you have an AA, those A alleles are what we call the non-secretors. So that would actually be, the ones that do not allow the B12 absorption from the gut into the bloodstream as readily or efficiently.
Mhm.
So, for those people, yeah, you may want to consider doing like a sublingual B12 supplementation if you're doing that, where the GG for the FUT2, those are the secretors. And then the other one was, what was it, MCM6 that she asked for?
I apologize. You're right.
Oh, okay.
It was, yeah,
Yeah,
MCM6.
MCM6. So the A allele is the optimal gene allele, and the G allele is the suboptimal for more of a lactose intolerance or partial lactose intolerance.
Mhm. All right. Jolene has our next question, and they're asking which gene is an indicator for leaky gut? Which result is optimal or suboptimal? Um, so, Jolene, leaky gut is a reflection of what is happening in the gut. It's really a statement of how is the biome and how are you supporting cells that should form that barrier between self and other. There isn't going to be one gene that is responsible for it. What I would offer, though, is you're not going to necessarily find the answer in genes. Genes will help inform the support and how you resolve it. But where you'd more pointedly find the answer is actually going to be in the Gut 360 premium testing, because that's going to look at that biome. It's going to look at that barrier. It's going to look at the stress or distress within the gut. There can also be some other testing through blood work, stool testing, etc., that can give a bit more insight into that. And so I'd encourage you working with a provider in order to assess it, because there's not really going to be one metric per se. It's going to be a collection of symptoms from clinical intake as well as some testing that will help discern that, and then genes inform the solution and the plan that help care for that. All right. Oh, I love this next question. Dr. Varden, do you mind if I steal it from you?
Absolutely, go ahead. And
Mm.
actually, that's I wanted to mention because I have my pups here and they just kind of like wanted to make themselves heard. But the one thing that with your cats, they like to be on screen, but at least they're quiet.
Every once in a while you will catch them purring. But you're right, they're not vocal insofar as they're not going to meow at you unless they're hungry, and then you will hear it. All right. Marianne has our next question, and the question is, how do you know if you will do well on a plant-based diet? Um, so Marianne, again, the answer is there's not one gene per se. There's going to be a collection of genes. Um, and I would look at or work with a practitioner to look at methylation. I would definitely look at FUT2 because that influences biome as well as B12, right? I would look at some of your detox genes, in particular ones that focus on gut. And I would also look at micronutrient status as well. Um, I will offer this. There are profiles that make it more difficult to be plant-based. There are profiles that would advise against it, right? Does that mean you have to adhere to that? No. It just means that if you choose to be plant-based and you have one of those profiles, you have suboptimal methylation, suboptimal FUT2, suboptimal a few other spaces, right? Suboptimal AME1, for example. um, that does mean that you're going to have to work really hard to be plant-based. So I'd really encourage working with a clinician to kind of do that self-assessment and then figuring out what is the best way forward. Because just because you can does not mean you should, right? Yeah. All right. Dr. Varden, do you want to, Sheila has a question for you.
Mhm.
How does zinc affect insulin resistance, and when is the best time to take it in?
Well, I will say that zinc does play a very important role in improving insulin resistance by supporting the insulin receptor function. Okay, it also helps in enhancing insulin action, reduces oxidative stress, helps to boost cellular glucose uptake. And studies have shown that adequate zinc intake is associated with lower blood sugar, improved insulin sensitivity, healthier lipid profiles, and of course can affect a decreased risk in type 2 diabetes. So specifically, zinc helps to synthesize, store, and release insulin from pancreatic beta cells where they're created. So it what it does is it helps to regulate insulin receptors. It helps with their sensitivity. And of course, zinc also acts as an antioxidant. It protects that insulin producing cells from the oxidative stress. But zinc also interacts with vitamin D and magnesium. And these nutrients are helpful by enhancing insulin sensitivity and metabolic health. So, when is the best time to take it? Well, optimal absorption of zinc is best when it's done on an empty stomach, generally like 30 minutes before a meal or about 2 hours after eating. But sometimes zinc can cause some nausea or stomach upset. So if that happens to you, if you are sensitive with that, it is okay to take it with food, preferably after a meal that's not rich in fiber, because the fiber can actually reduce the absorption of that zinc. So, I know some practitioners recommend taking it before bed to assist in tissue repair, possibly improving sleep, so it really kind of depends in that respect of, kind of test it out and see what works well for you. But honestly, consistency matters more than exact timing. So definitely taking your zinc daily for several weeks to see the impact on insulin resistance. And you also want to make sure to pair zinc with protein, not high fiber, calcium rich foods, because the protein could possibly aid in that absorption. Also be aware of the amount of zinc that you take, because if you're only taking zinc, it competes with the same receptors copper, and it can actually deplete your copper stores. And if you are taking, well, I mean, again, I definitely would speak with a provider to see the amounts that are appropriate for you. I'll just say that if you are going to be taking zinc, long term, make sure you have a milligram or two of copper in there. So that way you're not depleting those stores.
Hmm. Yeah. All right. Our next question is from Susan, and they're asking after um, what is uh, what is the optimal presentation of Amy 1 and how does that influence copy number variation? So, Susan, ideally, what you're looking for your Amy 1 is going to be seeing all T's. So TT, those are the alleles that confer kind of better management of salivary amylase and carbohydrate. If you see a sneaky little A in there, unfortunately, that will decrease your capacity to manage those, to produce that enzyme and manage it. In terms of copy number variation, we focus more on the SNP because we have strong research that really connects that to how much is released within the salivary glands, and that's where the research sits on: does it impact digestion? Does it impact metabolism and weight gain? So we actually focus on a SNP, and you'll find that within your diet and nutrition section. Okay. The next question I have here is from Carol. Um, they're asking if the gene outline, or so results from the DNA 360, um, says, let's see. It says, so if the advice is only having red meat a few times a week, and to cut out starches, and kind of like they have, it sounds like a diet and nutrition profile that says be careful of fats, be careful of carbohydrates, and be careful of protein. How do you actually get your protein? So Carol will offer a little insight into how we assess protein. Right now, we don't really have a gene that says you are good at digesting protein and you are not, right? And that's really what we're looking for when we're looking at protein. And that could be one of two reasons. One, we haven't found that gene, or one, it doesn't really exist because by and large, most people, based on how we've evolved, are great at digesting animal proteins, right? Um, and so, what we instead look at, because we don't have data that tells us any which way around protein, we actually look at more the inflammatory burden that they can come through, right? Because we know, unfortunately, based on North America, much of our animal proteins will come from factory farms, and they come from a lot of animal practices that don't necessarily translate well to the animal or to the people that consume them. They tend to have a lot of hormones, antibiotics. The feed might be more corn based as opposed to grass based. There's a few different variables in there. They can kind of come very laden on the plate. And that doesn't mention preservatives like the nitrates and the sulfates, etc. So we look at it more from a toxin burden. And so if it's saying be mindful of that, you actually more want to look at the quality of your meat and the cleanness of your meat. And then that will enable you to be consuming more, assuming obviously it fits well with your health picture. So that would be my thoughts and advice is there. Yeah. Dr. Varden, do you want to talk to? So Toya has a question for us. They're asking for thoughts on Akkermansia. So close,
Akkermansia, yeah.
so close,
And
audience.
Lana is the Akkermansia muciniphila is
Look at you and
generally
your fancy
the
syllables.
species the species that is in supplements,
Right.
and it has really gained a lot of interest for gut healing
Mm-hmm.
because of its role in supporting intestinal barrier integrity, metabolic health, immune modulation, because Akkermansia actually helps restore the mucus. That's where you get the mucinophilia, helps to restore the mucus barrier, which protects gut cells from toxins and microbes. It's also been shown to benefit in glucose control, weight
Mm.
management, reducing inflammation. It's actually one of the two species that has been found in research to upregulate GLP-1 naturally. That's the target of a lot of these semaglutides that are being given for weight control. And again, it helps by regulating immune responses, because some research has shown that it may lower intestinal inflammation. So, I mean, obviously there are still more research, safety trials, and other things going on, but Akkermansia is fairly well tolerated. Granted, it's not for everybody, not for people with active inflammatory bowel diseases or some post-infections, things like that. But there again, test, don't guess, see if where
Mm-hmm.
your levels are.
Yeah. 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 All right. Let's see. Kimberly has our next question asking if there's genetic code for protein in dairy instead of just the sugar. Not yet, but stay tuned in the weeks, months to come, maybe in the future. Yeah. All right. Jackie has our next question, and they're asking, can you speak on how eating organic slash non-GMO slash grass-fed affects this equation? Yeah. Dr. Varden, we knew that GST would come up amongst a few other things. Do you want to start tackle? I think this will take both of us, but do you want to start tackling this one?
Sure. And I mean, honestly, you also covered, quite a bit of this in the
Mm-hmm.
last question. When you were talking about protein, that
Mm-hmm.
it's not so much about the protein itself, because, and the reason, she brought up the grass-fed portion, but about the toxins
Mm-hmm.
that are there, that, how well are you able to clear these toxins? That really,
Yeah.
plays an effect. But one of the genes, working into that equation that Dr. Kosterman was just mentioning was the GSTM1, M as in Mary 1. And this really works into a detoxification capacity, because that gene codes for a key phase 2 detox enzyme. There we go. That's very essential for neutralizing toxins, and oxidative stress products. And again, this is a copy number variant. So you're either going to have 0, 1, or 2 copies. And this is the one you think, oh, 2 would be optimal. Actually, 2 can be a little over aggressive. One is more so your sweet spot. But this is where it can help with the anchoring of food and the microbes. So it definitely helps with the your microbiome, the integrity of that, the composition of that. If you've got an over-aggressive bouncer that wants to kick everybody out, you're going to have a little bit harder time keeping those buggies in place and
Mm-hmm.
having the food there, as compared to if you don't have a bouncer at all and all the riffraff coming in, then there could be problems. So
Mm-hmm.
it's really thinking about the GSTM1 like a cleanup crew,
Mm-hmm.
and without enough of those workers, the garbage, the oxidative stress, it tends to build up faster. So, you definitely want to make sure that you are feeding the good guys and then managing that appropriately. But it goes back to the toxins.
Mhm. All right. The next question is an anonymous question, and they're asking if we analyze genetic variants associated with poor or suboptimal pancreatic enzyme secretion. The answer is no, but I'm going to add that to my list of things to look into. So, not right now is the answer. Yeah. All right. The next question we have is um,
I know with Rachel. Okay, so this and I'm going to keep this succinct because we were just covering over the last few
Mhm.
questions. Rachel's asking, any idea why I can do most foods, breads, dairy, etc., in Europe with no problem, and then as soon as she comes home to the U.S., all of a sudden she reacts to the same foods. Reason? Toxins. Okay.
Mhm.
US allows a lot of toxins on our foods, pesticides, herbicides, all of these different things in our food systems. And this is where our bodies, it gets overwhelmed, where Europe and different places in Europe have much more stringent laws in use,
Kind of
toxins.
like lobbying. So I would say, Dr. Harden, what you're saying is, I wish it was still true. And I'd say in the past few years, unfortunately, that's changed. So, Rachel, I would say, like, if you've been to Europe recently, unfortunately, that's starting to shift. The lobbying body in Europe and many European countries is becoming more and more American. So we're starting to see this European protection, this mecca where those that can't do wheat here can do it over there, slipping. Interestingly, there's countries like Romania though that put up a huge temper tantrum when they threatened to bring that sort of stuff in, and it wasn't allowed. So there's a few. I think Dr. Varden, you were getting at this. There's a few little points within Europe that you can still get it, but it's interestingly
Right.
countries like Romania, as opposed to unfortunately countries like France or Italy, where they have historically prided themselves on the quality of food there.
Yeah.
Yeah,
And
which
it's
is
unfortunate. But again, this is where we need to know our detox
Mhm.
pathways, where we need support and just being careful.
Mhm. Yeah.
I think you need to answer Sandy's question.
Yeah. I'm all things gluten because it is banned from my body. How do you explain the HLA gene variants impact on gut health? Yeah. So if you are celiac, that is an autoimmune condition, and genes can predispose towards that risk. And so, if you are in that state of autoimmune, it is a self attack and it will erode and make difficult within the gut that lining, and it impacts many different things. In terms of sensitivity, what happens there is it creates low-grade inflammation, both within the gut, but then some immune byproducts that will travel elsewhere. And it sets the stage for leaky gut, amongst other things. Really, what it's doing is the inflammation reaction is wearing away at some of the tight gap junctions. So close to things that keep the cells together, and it makes your gut a bit more permeable. Okay, so it's kind of think about it like, especially if you have sensitivity, it's kind of like taking sandpaper and going into the gut and rubbing it, right? Every once in a while, if you have that sensitivity, some people can tolerate that, right? Some people can take a little bit of sandpaper every once in a while. But those that have other things going on in their gut, right? Maybe their biome isn't that great. Maybe they're eating other foods that are problematic to them. Maybe they're very stressed. Maybe there's another immune dysregulation. The body doesn't have the capacity to withstand or manage that sandpaper. So remember to take everything in context. And there's a lot of wisdom, especially if you're like, I want to walk on the wild side, maybe have some wheat, really listening to your body, because the impact of wheat for those who are really sensitive to it is in the moment, but also the half-life of some of the immune byproducts will linger in the body for like 8 weeks. Meaning, if you have that piece of that beautiful croissant today, for some people that are sensitive, in 8 weeks, you're still going to be feeling that impact. You may not know it explicitly, but that impact will still be there. So, just something to be mindful of for the wild walkers out there. All right. Let's see. Kristen, I can't see that far.
Well, we've got George who asked
Hmm.
a question.
Please.
We've got two more
Yeah,
questions
please.
left, which we have time for. So
Please.
George is asking, he said, just as proper gut bacteria is important to have for good gut health, he's heard that proper mouth bacteria is also tied to that. Absolutely true.
Mm.
So he's experimenting with swishing his mouth with unsweetened plain kefir, or kefir, however you want to pronounce it, then eating a piece of raw celery. Thoughts on this? And I love that you are informed about how oral microbiome is intimately connected to gut health. Oral bacteria can actually travel through the saliva and swallowing that to the gut, influencing the composition and behavior of the gut microbiome. Research has actually shown that a balanced oral microbiome promotes not just dental health, but also immune function and lowers that risk of systemic diseases, including metabolic and inflammatory conditions. And the neat thing is that kefir is rich in beneficial lactic acid bacteria and yeast species.
Mhm.
What I found interesting, because I learned this recently, that it's actually even more diverse than yogurt.
Mm.
So, I found that fascinating. But I know that some studies have demonstrated that regular consumption of that actually reduces markers of harmful bacteria.
Mhm.
Streptococcus mutans, not a good bacterium that you want, flourishing in the mouth. But it improves that oral microbiome. And, that's wonderful. And I love how he specified unsweetened plain. You definitely don't want to be having the sweetened flavored, those extra sugars. So that's very, very important. As far as
Mm.
raw celery, that actually kind of acts like a natural toothbrush. Chewing the celery can actually help stimulate saliva production, which helps to neutralize acids and wash away
Mm.
debris and bacteria. So, when you think about the fibrous texture, that's where you get that physical uh, promotion, acting like a toothbrush, because that can help disrupt biofilm. And of course, antioxidants that it
Mm.
has. So, I think that's wonderful. I mean, if it works, I actually personally haven't tried it, but theoretically and everything else, love to know how it's working for you.
Yeah. 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 That's interesting. Yeah.
Yeah.
Our last question is from Kristen, and they're asking if the DNA Gut 360 Premium can help with SIBO. They've noticed in particular that postmenopausal or perimenopausally, sorry, their gut health has been a lot worse and they're struggling with solutions there. Kristen, my ask would be if you could shoot us an email at clientcare@thednacompany.com. We'll put that in the chat in a second. Um, I'd like to get back to you personally on this one because my inclination is to say yes. I think between lower species as well as the perimenopause picture, there's going to be a lot of benefit. I'm just not, I need to look into the test if it's also able to peer into the upper intestinal tract, and I don't want to mislead you there. So if you send us an email, I'm going to find that out for you, okay? And I see that actually we want to put the client care email in there, Vanny, if that's okay. And thank you so much. All right. Audience, we made it. And hopefully you, your gut, and your buggies are all intact. Yeah. Dr. Varden, thank you for exploring this with me tonight. It's been a joy to explore the different genes and to explore how the buggies in our guts influence all the things. Yeah.
I love gut health and sleep and all those wonderful things. And I
Mhm.
really appreciated and enjoyed this conversation with you tonight. And I'm so happy all of you came and joined us to learn more about gut health and your genetics and how that all
Mm.
impacts your overall health. So thank you
Yeah.
and have
Yeah.
a wonderful night, everyone.
Yeah. All right. Au
I'll get
revoir.
to see you again tomorrow.
Yeah. All right. Au revoir, mes amis. Au revoir.
Uh,
Thank you so
Take care,
much.
everyone.
Au revoir.